Hello, my name is Dr. Alex Macknow, and I'm thrilled to welcome you to this lesson, Lesson 5.3, in which we'll explore the back half of the Kaplan method for science questions, the execute and answer steps. This lesson is a bit of a continuation on Lesson 5.2, in which we analyze the assess and plan steps. So if you haven't had a chance to watch that lesson, or if it's been a while since you've covered that material, it's probably not a bad idea to pause this video and go back to watch that lesson before continuing on with the execute and answer steps. In this lesson, we'll get to see the very real payoff of the plan step, as we execute on plans and choose the right answers. And that's what makes this lesson a lot of fun. It focuses on that moment on test day when you actually choose one of the bubbles and have selected the correct answer. That's a moment you'll have many times on the exam, and with the Kaplan method for science questions, we want to help you accomplish your goal of just sitting there on test day and churning out right answers, getting those points that you deserve. Let's start by delineating our learning goals for this lesson. There are two main ways to approach the execute step of a question. One is to predict a specific answer, which will then match to an answer choice during the answer step. The other is to think more broadly about the topics at hand, and then use process of elimination when looking at the answer choices to arrive at the correct answer. Both methods have their merits and flaws, and we'll explore those more during this lesson. We'll start by determining when predicting an answer is a useful strategy, as opposed to using process of elimination. We'll then work on making those predictions before approaching the answer choices, and finally, we'll work through the answer choices and recognize when an answer matches our prediction. The Kaplan method for science questions breaks the cognitive processes involved in problem solving into 4 discrete steps. The first 2 steps are to assess the question and plan your attack. If these first 2 steps are what you're looking for, then you can always watch or rewatch Lesson 5.2. But in this lesson, we'll be focusing on directly scoring you real points on test day. First, executing your attack plan for the question, which, as we'll see, is closely related to predicting an answer, and then moving through the answer choices themselves as efficiently as possible. The science topics in this lesson follow along with the central dogma of molecular biology, from DNA to RNA to protein and the processes therein. The focus in this lesson, though, will not be on the learning of this science, because that's covered elsewhere in your Kaplan resources, but rather applying this knowledge within test questions. So let's begin with the first learning goal. During this learning goal, we'll make that binary decision between making a more focused prediction versus thinking more generally about the question and using process of elimination. During this learning goal, we'll look at 8 question stems and make that same binary decision on each. First, a word of explanation on this exercise. You see 2 arrows for each question on your page. For a question where we would make a prediction and ultimately match that prediction to an answer choice, we'll choose the more focused arrow. Whereas for a question that we would think more generally about the science and use process of elimination to answer the question, we'll select the more general arrow. Take a look at this first question stem. This is a question for which a more focused prediction would be the appropriate response. The question stem mentions we have 2 fragments of DNA that separate, they come apart at different temperatures, and then we're asked about the differences between those 2 fragments with respect to their nucleotide composition. This means that the question is essentially asking us the connection between temperature, denaturation of DNA, and nucleotide composition. What would be the prediction for this question? Given the fact that fragment A melts at a higher temperature than fragment B, we would expect that fragment A is stabilized by a larger number of bonds than fragment B. This should immediately make us think about what types of bonds stabilize the connections between 2 strands of DNA, which are hydrogen bonds. The number of hydrogen bonds between different nucleotides differs depending on the base pairing we're looking at. Specifically, when cytosine and guanine pair with each other, they form 3 hydrogen bonds to stabilize the connection between the 2 strands. On the other hand, when adenine and thymine pair together, they only form 2 hydrogen bonds. Given that information, we would expect that fragment A is stabilized by more hydrogen bonds, and therefore likely has more cytosine-guanine pairs in comparison to fragment B. How about this question? This is a very broad question. We're asked for any statement that's true about transcription, so more general thinking about the process would be appropriate before jumping into the answer choices. There are many possible specific predictions you could make for this question. You may already be thinking about some of the characteristics of transcription, such as the production of hnRNA, which is later processed to mRNA from a DNA strand, the fact it occurs in the nucleus, that hnRNA is produced from 5' to 3' end, that RNA polymerase II is chiefly involved in this process after binding to the TATA box in the promoter region of a gene, or aspects of post-transcriptional processing, like the 5' methylguanine cap, 3' poly A tail, or splicing together of exons, or many other things. Now, if all those don't come to mind at this very moment, then that's probably a signal that you could use more content review on the subject between now and test day. But the main takeaway from this question is that this question, along with those that look similar to it on test day, is not one for a strong prediction. Therefore, we should use the elimination strategy for a vague, open-ended question like this one. If you're eager to try that strategy out yourself, don't worry, we'll get to do it several times before the end of this lesson. How about question 3? Take a moment to read through this question stem and determine the type of prediction you would make, a more focused prediction or more general thinking about the question. If it's difficult for you to tell if you could make a specific prediction for a given question stem, here's a good trick. Think about if someone walked up to you on the street and asked you that question. Would you be able to give a specific answer? If so, making a specific prediction is probably the best way to approach that question. If not, or if you feel that there would be multiple possible answers, then thinking more generally about the question before using process of elimination is probably the better technique. This is a question for which more general thinking about the science at hand would be useful. There are many possible reasons for an elevation of mRNA within the cell, but all of them would center around the same theme, which would be an appropriate prediction here, that the cell is being signaled to create more protein. But that signal could come in many forms. The cell could be receiving autocrine, paracrine, juxtacrine, or endocrine signals. It could be responding to its environment or receiving neurological signals. Given the fact that there are many possible specific predictions we could make here, it actually behooves us to think more generally about the science and to look for an answer choice that fits into that theme, that the cell is being signaled to create more protein because mRNA would later be translated to form that protein. Let's check out question 4. Read through the question stem and again determine the type of prediction you would make for this question, a more focused specific prediction or more general thinking about the science at hand. This is a question, again, for which more general thinking about the science would suffice. It's another vague, open-ended question here, although the science is called out relatively clearly in the question stem. We're looking at DNA methylation, meaning that an appropriately general prediction here would be thinking about what we know about DNA methylation and then using process of elimination. If we're familiar with DNA methylation, we know it's one of the main inhibitors of gene transcription, so we would look for an answer choice about turning genes off. Let's look at question 5. Again, read through the question stem and determine the type of prediction you would make, a more focused prediction or more general thinking about the science at hand. This is a great example of a question for which we can make a very focused prediction. We should be able to come up with the exact answer. In the question stem, we're told that during PCR, or polymerase chain reaction, DNA is unwound by using heat instead of DNA helicase, and we're asked what bonds must be broken to unwind that DNA. Well, the bonds that hold DNA strands together are hydrogen bonds. So to separate two DNA strands or denature the DNA, we would have to break those hydrogen bonds to break it into two separate strands. Let's now look at question number 6. If you've completed lesson 5.2 recently, you've actually seen this question before. It was question 2 in that lesson. Read through this question stem and again determine the type of prediction you would make, a more focused prediction or more general thinking about the science at hand. This is a question for which this strategy is key. While it is possible to come up with the exact RNA transcript that we would get from this strand of DNA, it's actually a more efficient approach for this question to use more general thinking about the science to arrive at the answer. Why is this? Well, given the topic at hand, there are certain classic traps we might expect to see in the answer choices. Specifically, because we're talking about DNA to RNA transcription, we might expect to see answer choices that are not RNA but DNA instead. We might also expect to see incorrect answer choices that have the polarity in the incorrect direction, writing the transcript from 3' to 5' end instead of 5' to 3'. Finally, we might expect incorrect answer choices that arise from using base pairing incorrectly or not remembering what the base pairing rules are. So the best prediction for this question would be keeping in mind that we'll get rid of any non-RNA answer choices, specifically any answer choices that have thymines in them instead of uracil, and then thinking about the first 4 base pairs and seeing if that's sufficient to answer the question. Now, that may not sound like it's saving a lot of time rather than writing out the full transcript, but even if you're only saving, say, 5 seconds per question by thinking about it strategically, well, 5 seconds times 59 questions, you're saving just about 5 minutes on the entire section, which is time that you'll want to be able to spend answering more questions, answering harder questions, or getting to go back and check your answers to previous questions. You can see these small gains in time certainly do add up, so using these strategies to maximize time saving while still arriving at the correct answer is key on test day. Let's move on to question 7. Read through the question stem and again determine the type of prediction you would make, a focused, specific prediction or more general thinking about the science at hand. This is a long and somewhat convoluted question stem, and it doesn't give us quite enough information to come up with an exact focused prediction, so our best approach here will be more general thinking about the science at hand. Again, we'll use the elimination strategy here, but it's important to discern what exactly we'll be looking for in those answer choices to help us eliminate incorrect answer choices and arrive at the correct one. The question stem tells us that the different blood types are actually heritable through genes, but that the difference between blood types is not a difference in DNA or protein, but actually a difference in carbohydrate that's attached to a common molecular backbone. So the underlying question here, as it's addressed at the end, is trying to explain how a gene could determine something besides protein and rather account for differences in carbohydrates. We'll be looking for an answer choice that simply links the idea of DNA and genetics to carbohydrates that could exist in or on a cell. Let's move on to the last question of this learning goal. We haven't answered any of the questions on this page in full yet, but this final one will be an exception. We'll bring it all the way to the answer now and use it to highlight Kaplan's Roman numeral strategy, which you'll almost certainly use on test day. Take a moment to read through the question stem and determine the type of prediction you would make, a more focused specific prediction or more general thinking about the science. With most Roman numeral questions, you'll actually have to do a bit of both in this case. We will want to make as specific a prediction as we can, but then we'll use that prediction, in tandem with the Roman numerals provided here, to use process of elimination to arrive at the answer as quickly as possible. What would your prediction be for this question? We're told in the question stem that a person has a mutation in the promoter site of a gene, which makes that promoter site non-functional, and we're then asked what symptoms would occur. If there's a non-functional promoter site, then RNA polymerase II will not be able to bind to the DNA strand, and transcription will not occur of this gene. If transcription of the lactase gene does not occur, then the lactase enzyme will not be formed through translation, and we would expect this person to have no functional lactase. If there's no functional lactase, we would expect that person to be unable to digest lactose as well. With that, let's work through the Kaplan Roman numeral strategy. Inexperienced test takers will often approach Roman numeral questions by analyzing all of the Roman numeral statements before hitting the answer choices. This is inefficient, however. After all, what happens if, on a question like this one, you end up with the combination 2 and 3 and that's not an answer choice? When that happens, you have to go back, do more reanalysis of each of the statements, and you lose a lot of time. So in the Kaplan Roman numeral strategy, we analyze one of the statements to determine if it's true or false and if it answers the question, and then knock out answer choices that do not fit that prediction. Oftentimes, in doing so, you won't have to check all of the Roman numeral statements because you'll arrive at the correct answer earlier than that. Let's start with Roman numeral 1. This one's probably the easiest to start with because it flows most logically from the thinking we've already been doing on this question. This statement does quite cleanly match our prediction. If a person no longer produces the lactase enzyme, they will be unable to digest lactose in the gut, meaning that statement 1 must be true, and we can cross out answer choice B using process of elimination. Let's move on to Roman numeral 2 because it still addresses digestion. Roman numeral 2 does not quite match what is stated in the prediction, but still makes sense as a sensible answer. If a person does not digest lactose him or herself, well, that lactose can still be broken down by the symbiotic gut bacteria that exist primarily in the large intestine. This is actually part of the explanation for why individuals with lactose intolerancy, which can be due to lack of the lactase enzyme, often get flatulence and diarrhea as symptoms when they ingest dairy products. So in this case, we would expect Roman numeral 2 should show up in the answer choice, and we can actually knock out answer choices A and D, leaving us with the correct answer, answer choice C. Notice that the Kaplan Roman numeral strategy makes it so we actually don't have to check all of the statements that are given in the question stem. In this case, we didn't even have to check Roman numeral 3, and yet we know we've arrived at the correct answer. But for the sake of completion, let's still look at Roman numeral 3. This statement implies that lactose would be the only source of glucose for this individual. Indeed, lactose is one source of glucose. It's a disaccharide made up of glucose and galactose, but there are many other sources of glucose in the diet, including sucrose, another disaccharide made up of glucose and fructose, as well as starches and simple glucose monomers. So we would expect that this individual, even if he or she cannot digest lactose, would not have a glucose deficiency because there are many other sources in the diet. So we've looked now at the difference of when to make a more focused prediction versus when to think more generally about the science. But as the Kaplan tip on the bottom of your page points out, there is a third option. When neither of these two options seems like a good choice, or when it's difficult to come up with a prediction, we can always use strategic guessing to arrive at an answer. But use strategic guessing, not random guessing. Knock out answer choices that you know to be incorrect, and then eliminate other answers for the best reasons you can think of. So now that we've looked at when to make a more focused prediction versus when to think more generally about the science, let's actually start working on the execute step and coming up with those predictions. We're going to do this in the context of a passage, and this is actually the same passage that was presented in Lesson 5.2. So if you've seen Lesson 5.2 recently, feel free to skip ahead to when we actually start executing our plans for these questions. If it's been a while or if you haven't seen Lesson 5.2, take 5 minutes to read and outline this passage, and then I'll go through the passage, pointing out some of the salient points from each paragraph and putting together a Kaplan passage outline for it. Starting with the first paragraph, we're introduced to our topic for this passage, which is Hutchinson-Gilford progeria syndrome, or HGPS. This paragraph mostly serves as an introduction, presenting some of the signs and symptoms associated with this disorder. Like many MCAT passages, the first paragraph serves mainly to introduce us to our topic, but doesn't really have a lot of testable information. An appropriate label for this paragraph might be that this is our introduction to HGPS, and that we're given the signs and symptoms of this disorder. In the second paragraph, we're given quite a bit of information about the molecular biology underlying HGPS. We're told that a point mutation occurs in this disorder in the gene for lamin A, and that this point mutation ultimately causes a shortened mRNA transcript to be generated. Once this mRNA transcript goes through translation and moves into post-translational processing, it means that it can no longer lose this farnesyl group, which ultimately causes it to be locked to the nuclear rim. Notice that they give us a new term in this sentence, they've defined it in the previous one, that this mutant prelamin A is called progerin. We're told that this locking of progerin to the nuclear rim means that it can no longer offer the structural support that it needs to the nuclear envelope, and that ultimately this causes a misshapenness to the nuclear envelope, and that down the line it's implied that this causes issues during mitosis. Well, that's a lot of information, so if we distill that down to a short label for our passage outline, we could say that the cause of progeria ultimately is this point mutation causing a shortened mRNA transcript. For the rest of the details, we can always come back to the passage and locate them. For now, this should suffice as an appropriate description for the paragraph, and we'll keep moving forward. So if paragraph 2 focuses on the aberrant molecular biology that occurs during Hutchinson-Gilford progeria syndrome, it should be no surprise that the third paragraph contrasts this with normal prelamin A post-translational modification. We're also told that this happens in 4 steps, so let's trace through these 4 steps one by one. The first step is the farnesylation of a cysteine residue in the protein by an enzyme called farnesyltransferase, or FTase. We're then told in the second step that a small portion at the carboxyl end of this protein is removed. And then, for the third step, that product is methylated. Finally, for our fourth step, a larger portion of the protein is cut off by a peptidase called ZMPSTE24. We're given a reference to a figure, which will be helpful to be able to visually demonstrate what happens during this process, and at the very end of the passage, we're given a quick note that FTase inhibitors appear to reverse some of the malformations illustrated in paragraph 2, and therefore may be a potential therapy for this disorder. There's a lot of information in this paragraph, so again, it's not worth writing all of it down, but an appropriate label for this paragraph would be noting that it's illustrating the process of normal post-translational processing for pre-lamin A. Finally, we're given two figures which illustrate the process of post-translational processing in Hutchinson-Gilford progeria syndrome, or with mutant pre-lamin A, as well as during the normal process. It's important to note that these figures include almost all of the information from paragraph 3 and quite a bit of the information from paragraph 2 as well. If we need to come back to the passage, these may serve as a quicker source of information than reading through all of the text. While labels are provided for the figures on the MCAT, it's still important to put labels into your outline for each passage element. So, for the first figure, we might note that this is HGPS post-translational processing, whereas for the second figure, it's focused on normal post-translational processing. Now that we've worked through the passage, let's turn our attention to the questions. Let's start with question 9. An appropriate plan to attack this question would be to go back to the passage and determine what FTase, or farnesyltransferase, does in progeria. There are a few sources of information from this. Paragraph 2, paragraph 3, and the figures actually all deal with farnesyltransferase in some way. But the most appropriate place to go of those is likely figure 1. It gives us a visual representation of the molecular biology of progeria, so that's an appropriate place to start. Take a moment to look at figure 1 and determine what your prediction would be here. Why is it that when FTase inhibitors are used in these animal models that the malformation of the nuclear envelope may be reversed? Taking a look at the figure, FTase is pretty easy to spot. It's the initiator of this process, the first enzyme that acts during post-translational processing in Hutchinson-Gilford progeria syndrome. So if this enzyme were blocked and no longer permitted to act, we can see that this process just simply wouldn't occur. Without that first step, we would no longer have progression through the entirety of post-translational processing, and we would no longer get progerin as an end product of this process. So an appropriate prediction for this question would be that these FTase inhibitors actually stop the production of progerin. It was progerin that caused this malformation of the nuclear envelope, so we can imagine if progerin is simply not being produced, that we would no longer see this malformation, and it would appear to reverse not only in animal models, but perhaps in humans as well. So with that, we're armed with a strong prediction. We're going to look for an answer choice during the answer step that basically says that we would no longer get progerin produced if FTase inhibitors are used. Let's move on to question 10. A good plan for this question would be to go back to paragraph 3. This question is asking for the amino acids at the carboxyl terminus of pre-lamin A, the normal processing of which is described in paragraph 3. So, take a moment to look at paragraph 3 and determine what your prediction would be for this question. What would we expect for the amino acids at the carboxyl terminus of pre-lamin A? The paragraph describes these amino acids as being aliphatic amino acids, which means we'll have to call upon our science knowledge to determine what exactly they mean. An aliphatic compound can be contrasted with an aromatic compound. Aliphatic compounds are non-aromatic, they're often described as fatty, usually composed of carbons and hydrogens, and they do not contain conjugated ring systems the way that an aromatic compound would. So an appropriate prediction for this question would be that we're looking for an aliphatic amino acid, or in other words, an amino acid that does not have an aromatic group. We might expect that the wrong answers, by the way, all do contain this aromatic group, which would allow us to use process of elimination to choose the correct answer. How about question 11? An appropriate plan for this question would be to go back to the passage and look at paragraph 2. We're being asked about the underlying mechanism or molecular biology underlying progeria, so it makes sense to go back to this paragraph and find what the mutation is that occurs in progeria and how that might cause the issues that occur in progeria. Take a moment to look at paragraph 2 and determine what would your prediction be for this question. What is the etiology that underlies progeria? While there's a lot going on in this paragraph, the underlying cause of progeria is actually made quite clear. We're told that it's a point mutation in a gene coding for lamin A, and specifically that the mutation is a change from cytosine to thymine, and that this causes a shortened mRNA transcript to be generated. So an appropriate prediction for this question would be that this point mutation causes a shortened mRNA transcript. Have some caution when we move into the answer choices. Whenever dealing with molecular biology and the processes of transcription and translation, it's important to keep these processes straight. Expect that wrong answer choices here may allude to translation instead of transcription, especially because the passage seemed to focus on post-translational processing, although the underlying mechanism here is actually a problem during transcription. Let's take a look at question 12. Given that the question stem mentions some of the signs and symptoms of Hutchinson-Gilford progeria syndrome, as mentioned in paragraph 1, a good plan would be to think about the causes and associated features of progeria. So what would your prediction be for this question? This is a question where our prediction should stay relatively broad and we should use process of elimination. Again, the question is asking what causes or is associated with progeria. We may be thinking about the point mutation and the underlying molecular biology that causes progeria, or we also might be thinking about the associated microscopic signs that are described in the passage. All of these would be potential candidates for answers. For a question like this that again is relatively vague and open-ended, we shouldn't predict a precise answer, but should rather walk into the answer choices armed with the idea that we're looking for something associated with progeria. By extension, wrong answer choices are probably going to be characteristics of healthy individuals or completely unassociated with the disease altogether. Finally, let's look at the last question. A good plan for this question is first recognizing that this is a pseudo-discrete question. We don't need to go back to the passage for information. And then thinking about the differences between cytosine and thymine. So what would your prediction be? What are the differences between these two nitrogenous bases? Well, the key difference between cytosine and thymine are their partners. In other words, who cytosine pairs with, that's guanine, using 3 hydrogen bonds, and who thymine pairs with, that's adenine, using 2 hydrogen bonds. So we would expect that this change from cytosine to thymine reduces the number of hydrogen bonds that exist in this molecule. We wouldn't expect a difference in terms of the chemistry, at least purine versus pyrimidine, because both cytosine and thymine, and uracil for that matter, are pyrimidines. These nitrogenous bases can be remembered with the mnemonic cut the pie. CUNT, cytosine, uracil, and thymine are all examples of pyrimidines in terms of their chemistry. As we wrap up this learning goal, it's worth taking a look at the Kaplan tip on the bottom of your page. As it points out, the prediction that you make during the execute step is valuable whether you're dealing with an easy passage or tough passages. Even if the prediction we make cannot be precise, making a prediction at all and thinking about the science at hand gets us thinking in the right way as we walk into those answer choices. It will help us avoid being distracted by trap answer choices, and even if we have to use strategic guessing at the end, creating that prediction often makes it easier to knock out some of the answer choices, improving your chances of guessing the correct answer.
Hi there, my name is Adam Gray. Sorry for the change of pace a little bit, but I'm going to be subbing in for Dr. Alex for the last learning goal of this lesson. Thank you very much to Dr. Macknow for taking us through so excellently through the first 3 steps of the attack strategy. Now let's hit the 4th. This learning goal is all about what we all been waiting for, actually taking our prediction, whether it be focused or general, and matching it to an answer choice. And so the verb here is really recognizing. You're going to have a strong thought in your mind, and we'll review the prediction that we've already come to, and the exercise here will be to just match it to an answer choice. We are starting here with question number 9, which is the one that asks more or less why FTase inhibitors work to reverse the effects of progerin. And our prediction was that they stop progerin production in the first place. So go ahead and pick that answer. What choice matches this prediction? The correct answer is choice B, matches our prediction pretty quickly, and this is a great example of how quickly you can answer questions that might otherwise be complicated if you have that strong prediction in mind. And of course on test day you would just move on to the next question at this point, but for this and the other questions we're also going to look a little bit at the wrong answers because they're interesting. They play with the strategy and especially with the science in some interesting ways. In this question, choice A is wrong because it says that these FTase inhibitors themselves would catalyze the cleavage of the farnesyl group. Remember, the modified farnesyl group was what caused all these symptoms in the first place. And certainly, if FTase inhibitors did catalyze that cleavage, that would be nice, but that's not what they do. In fact, we're given an enzyme in figure 2 of the passage that does this upstream cleavage. It's ZMPSTE24. The FTase inhibitors are actually going to work in a much earlier step of the process, so that upstream cleavage never gets a chance to happen or not happen. Choice C is wrong because, again, it's a little too optimistic about what these FTase inhibitors do. Remember that Hutchinson-Gilford progeria syndrome is a genetic disease. No amount of drugs that we give a patient, unless we're giving them some kind of genetic therapy, is going to give them the alleles that allow them to code for the correct protein, for the normal prelamin A. And so the FTase inhibitors are not going to be able to snap their fingers and just fix everything. Again, what they actually do is prevent any prelamin A from being created, either defective or normal. And then finally, choice D is wrong because, like choices A and C, if the FTase inhibitor did this, it would in fact treat HGPS. If these inhibitors physically prevented progerin from locking to the nuclear rim, then that would relieve some symptoms, because the passage told us that the symptoms come from the fact that progerin locks to the nuclear rim. But again, that's just not how FTase inhibitors would work, given the information that we know in the passage. FTase inhibitors work way earlier on in the process. They prevent the defective prelamin A from being created in the first place, so it's not even there to lock or not lock to the nuclear rim. Note again how clever all these wrong answer choices are. These are things that would reverse the malformation of the nuclear envelope in all cases, but only one choice, choice B, is the one that actually matches what the FTase inhibitor would do. Now let's move on to question 10. This was the short question where the answer choices are all amino acids, and our prediction was that the amino acid, the correct one, must be aliphatic. So go ahead and answer. Which choice matches this prediction? I hope you had your amino acids memorized, or roughly so. The correct answer here is going to be alanine. All three of the other answer choices, tyrosine, tryptophan, and phenylalanine, have some kind of aromatic group, and they're therefore not aliphatic. Alanine, on the other hand, just has a simple methyl group, which makes it non-aromatic. Notably, even if you couldn't recall the structure of all these amino acids right by looking at them, which, mind you, by test day, you do need to know all 20 of the amino acids, but if you didn't hear, there's at least a little bit of a hint in that choice C says phenylalanine, which is a good hint that there is in fact a phenyl group, or an aromatic ring, on phenylalanine. So you could, even with just that hint, eliminate choice C and allow yourself to guess a little bit better. But ultimately, you should use your knowledge of amino acids to get to answers like this. Now on to question 11. A long question with long answer choices, so it's a good thing that we have a nice solid prediction. We followed the information in the passage to say that the reason faulty prelamin A gets produced is because there's a point mutation that leads to a short mRNA transcript. Now that you can look at these answer choices, pick the answer choice that best matches this prediction. And the best match here is A. It doesn't come out and match our prediction perfectly. It doesn't say shortened mRNA transcript, but it does say that there is a loss of RNA from one of the exons in prelamin A. Drawing on your knowledge of molecular genetics, remember the exons are the parts of the RNA that get expressed. In other words, the exons are the ones that get left on the mRNA transcript. If we lose RNA from one of those exons, then we're going to have a short mRNA transcript. And the mechanism suggested for this by choice A, that the mutation introduces a new splice site, is a perfectly plausible way this could happen. During post-translational processing in the nucleus, if this hnRNA is cut one extra time, then you might end up with less RNA. The wrong answer choices here are also very interesting though. Especially if you picked one of these, you're going to want to know exactly why they're wrong. Choice B is wrong because it's about the wrong process. Choice B says a stop codon will appear a little earlier, but an extra stop codon wouldn't shorten the mRNA. The extra stop codon would shorten the protein. Choice B is an answer that's talking about translation into a protein, not transcription. And long ago, he determined this was an error in transcription or post-transcriptional processing and not an error in translation. Choice C is wrong because not only does it not match our prediction, that would be enough of a reason to eliminate it, it is directly contradicted by the passage. The passage says all that happens here is a cytosine is replaced with a thymine. It's a point mutation. If the DNA was spliced and reformed, that is, completely cut and put back together in a different way, that's a lot more than a point mutation. And that's not what's happening here. And finally, choice D is wrong because, again, it doesn't match our prediction, but what makes it clearly incorrect is that we have no evidence that this faulty prelamin A is any less stable as a result of the mutation. In fact, we have a pretty good hint that it's really stable, given that what this faulty prelamin A does is lock on to the nuclear rim and not let go. It probably is pretty stable because it leads to a lot of symptoms over time. Choice D might be correct if this was a disease that came from a missing protein, but that's not what this disease is. Hutchinson-Gilford progeria syndrome is a disease that comes from too much protein. After this faulty prelamin A is produced, then it starts to cause problems. Therefore, the problem would be that it would be very stable, not that it would be less stable. So now let's move to question 12. This was the one where we made a pretty vague prediction. We know we're going to have to eliminate answer choices because the question mentioned some symptoms of progeria, stunted growth, and musculoskeletal degeneration, and asked which is going to cause it. So really the question is, which one is caused or associated with progeria? So we need to use process of elimination, pick the answer choice that does cause progeria, and get rid of the ones that don't have anything to do with progeria or don't cause progeria. Given that prediction, as general as it is, which answer choice matches it? And the answer here is choice D. This question is a perfect example of why we can't expect to have a really focused prediction for every question. Even though D is the correct answer, we haven't seen anything before, at least not in this passage or this question, about manipulation of chromatin during mitosis. But it is the only one of these answer choices that is likely to cause the symptoms of progeria. Specifically, that's because it's the only answer choice that is pathological at all. All the other choices, A, B, and C, are what happens in a normal situation. Choice A is wrong because, well, that sounds fine. If you have a structurally supported nuclear envelope, then that's great. There's no reason why you'd have stunted growth or anything else bad. In fact, what Hutchinson-Gilford progeria syndrome does is mess up that usual structural support of the nuclear envelope. So A is the opposite of true. B is wrong too because it is what happens normally. If you compare the two figures from the passage, remember figure 1 was the abnormal processing, figure 2 was the normal processing. You saw that farnesyl group fall off in number 2, in the normal processing. Again, the problem with progeria is that that farnesyl group doesn't get lost. And then C is wrong for the same reason as A and B. We learned from the passage and the figures that methylation is a part of the normal processing. It isn't part of the bad stuff that happens during HGPS. And now let's bring it home with number 13. The prediction here was, what are the differences between cytosine and thymine? Well, we've got different pairings, but both are pyrimidines. That's not a difference. Given this prediction, match to an answer choice now. And the answer here is A. We can see from this prediction, clear as day, that one fewer hydrogen bond is formed between base pairs if you change from a cytosine to a thymine. C and G make 3 bonds, A and T only make 2 bonds. Choice B is wrong because, as we happen to write in our prediction, a pyrimidine is not changed to a purine. Cytosine and thymine are both pyrimidines. You can remember that by the good old mnemonic cut the pie, meaning cytosine and thymine, as well as uracil, are pyrimidines. Choice C is wrong because this change, putting an OH group onto deoxyribose, would be changing DNA to RNA. That's not happening here. And then finally, choice D is wrong because, again, we don't have any evidence that this is happening. The backbone of DNA gets deformed when you have a base pair mismatch. But here, since the problem is a point mutation, this mutation was there long before. It may have even existed before fertilization in the gametes. And that means that the incorrect nucleotide, the thymine instead of a cytosine, is going to have a matching base pair on the other side. Because once you have a point mutation, then during DNA replication, that point mutation is going to perpetuate across all future copies. Now, mind you, choice D is a thing. Phosphate backbones occasionally do get deformed when DNA is damaged due to, for instance, ultraviolet radiation, but that's a very different subject from point mutation. It comes up more in situations of DNA damage and repair. And that means that we've now gone through the entire journey for all 5 of these questions connected to the passage. As the Kaplan tip at the bottom of your screen says, make sure to not forget your prediction as you move to the answer choices. You can think of your prediction as kind of a shopping list. If you know that you're going to the store and you need to buy milk and eggs and flour and sugar, then you bring that list to the store, you pick things up, and you get back home really quickly. But if you forget your list when you go to the store, then you're going to be unsure and you're going to be searching all the different aisles and it's going to take you a lot longer. That's exactly how predictions work on the MCAT. And that should be exactly what you're taking away from this lesson. Speaking of your takeaways from this lesson, specifically it was about steps 3 and 4 of the Kaplan method, or the attack strategy, depending on what you'd like to call it. The execute step is when you are executing the mental plan from the plan step. It usually involves going to different places for information, the passage, your mind, your content knowledge, the question stem itself, and synthesizing it all together into some kind of a prediction. We saw in the exercise at the start of this lesson, sometimes a more focused prediction that you just try to match to an answer choice, sometimes a more general prediction where you refresh yourself on the science and then try to find the one that makes the most sense. Speaking of what you do once you get to the answer choices, depending on the type of prediction you made, you'll do slightly different things when you go to the answer choices. If you made a solid focus prediction, you'll probably just try to match an answer choice with that. Your prediction will be something like, remember from question 9, FTase will prevent progerin from being made, and then you just find the answer choice that says FTase prevents progerin from being made. On the other hand, if you made a general prediction, you can use process of elimination with that general prediction as a guideline. Remember from question 12 here. Our general prediction was we need an answer choice that aligns with progeria. And there was only one choice that did that. We eliminated all the ones that didn't. There's also one other way you're going to be moving to answers on test day, and that is strategic guessing. For instance, when you need to move really fast through a question or you're really not sure of the science behind it, you'll need to just eliminate what you can and make your best guess. We didn't do much of that here, but there are other places for you to learn and practice strategic guessing in your Kaplan program in later lessons, and you will get the chance to hone those skills there. And that brings us to the end of yet another lesson. Use the questions that we completed here as a guide for how well you know this attack strategy so far. Did you get those 5 questions right as you were matching them, or did you still get some wrong? If you did, then try to diagnose why and continue to practice. Also keep in mind that even though we really only did 5 major questions in this lesson and it took us a while, that's how learning works. You do things very slowly at first and gradually, picking apart every one of the steps. Then you move faster and faster as you build mastery, and as this method becomes more of a second nature. The reason why we use this method is it's because it's how expert MCAT test takers answer MCAT questions. So that's exactly how you're going to answer them as you build your own expertise. Thanks a lot. Good luck on your test.